Today I'm going to be talking about single cell models for state-dependent eQTL analysis. This is something that I started thinking about during my PhD in Shomo Raychaudhuri's lab at the Brigham and also here at the Broad. But it's a field that's changed a lot and grown a lot even since a couple of years ago when I was working on some of the work I'm going to share today. So today's primer is essentially going to overview kind of what's happened, what's changed in the past couple of years, and where we still have room for improvement in these models. So I think a lot of people talk about how single cell studies are growing, they're getting bigger, we have more cells, we have more donors. But I think a specific aspect of this that's really interesting is the fact that we also now have bigger single cell studies from genotyped cohorts. And so there are a couple of really big examples of this published last year in Science, one of which is the paper shown here, which had almost 1,000 genotyped individuals and single cell RNA sequencing of PBMCs or white blood cells from these individuals. We've also done sort of similar kind of study designs where we published a paper in 2021 looking at 259 individuals and now doing not just RNA sequencing, but site-seq, so single cell RNA and protein expression. And there's also studies emerging, although there are definitely few of them, in cell types that are not blood or not PBMC based. So there's a lot of iPSC studies now looking at iPSC cell lines differentiated into different cell types from a large number of genotype donors. And to kind of start to give you a visual of this, this plot is essentially showing different studies that had these two types of data, single cell RNA sequencing and genotyping or whole genome sequencing. You can see that they're really only starting to emerge in the past few years, and they're getting pretty big pretty quickly. And so it's a really interesting type of data to work with because it allows us to do the types of studies that link genetic variation to single cell phenotypes. So probably the most vanilla version of this is you could imagine having a variant, having some position in the genome where you could either have the A allele or the T allele, and then maybe if you look at your single cell phenotypes, you see some kind of phenotypic difference between people who have A or T. So we can do this, even without single cell data. So where does single cell data push us forward here? Well, it allows us to look for phenotypic changes that might be cell type or cell state specific. And so, this is something that single cell data offers that bulk didn't. The problem is to actually find these types of associations, we also need statistical models that can work at the single cell resolution. And so as I've kind of alluded to, today we're really going to focus on eQTLs. There's lots of different phenotypes we could look at, but with eQTLs, we're focusing on genetic variants that are somehow associated with gene expression. So you might have two different alleles for a given gene, and then maybe allele A leads to lower expression, allele B leads to higher expression. And the way that we typically visualize this is with some kind of box plot or some other types of plots, where we can see that as the number of copies of the alternate allele increase, the expression of this gene is increasing. When we do this in bulk, we can typically find these associations using some kind of linear regression tool, FastQTL, TensorQTL. These are both pretty common tools that people use. And what these tools will do is they'll try to fit some kind of linear regression and calculate a beta that represents the change in the expression of this gene for every additional copy of the alternate allele. So this works in bulk. And for a long time, this is also what we were doing with single cell data. We were doing what we would call pseudo-bulk eQTL mapping, where we essentially take a single cell data set and transform it into something that looks a little bit like bulk. And this flowchart kind of shows two main paths that people have followed to get to the pseudo-bulk state. One option is you can just take your single cell data for all the cells from a given sample, just add up their expression of each of your genes. Then you end up with essentially a genes by samples matrix. Then you can normalize that the way you normalize normal bulk data. The other path you could take is actually just normalizing the single cell data the way you would work with single cell data. So with SRAT, with SCRAN, whatever you're using. And then instead of summing up the expression across the cells from each sample, you take the average of the expression of each gene for the cells from each sample. Whichever route you take, you end up with some kind of normalized samples by genes matrix. And what can you do with that? Well, you can actually just put that right into your bulk eQTL mapping pipeline. And so this is what most of those studies I showed on that plot that have come out in the past few years, most of them have taken this approach, which is the pseudo bulk eQTL approach. But of course, this has limitations if you actually care about sort of single cell resolution effects. The main limitation that I'm going to highlight today is the fact that these models don't account for the heterogeneity of single cell states. And so when I think about a single cell state, we think about these eQTLs that could be different based on what cell type you're looking at, or what cell state you're looking at. So I'm going to mention cell types and cell states a lot in this talk. When I say cell type, I'm typically talking about things that are independent, separate lineages. And so maybe a macrophage versus a T cell. You could imagine that if you did eQTL mapping in a macrophage, maybe at one particular allele, you see no effect on the expression. Maybe if you look in a T cell, you do see that eQTL. And this is probably mediated by some type of cell type specific transcription factor, some type of cell type specific regulatory element. It could even get even more granular. So maybe I just want to look within T cells, and even within T cells, I might be able to see differences in whether an eQTL is present in one type of T cell state versus a different T cell state. So memory versus naive. So if we want to find these types of eQTLs, we can't just use pseudo bulk, throwing all of our cells in, combining all their expression. But you might say, wait, no, I've read these studies. They are able to look at this type of cell type specific or cell state specific expression by looking at the clusters. So you can take your single cell data, you can cluster it, and then you can essentially do pseudo bulk eQTL analysis within each cluster. And so it's the same process I mentioned before. The only difference is rather than starting off with all of your cells, you can break it down into different matrices, one for just T cells, one for just macrophages, and then again, turn those into samples by genes matrices with pseudo bulking. And then you just do a T cell specific eQTL analysis or a macrophage specific eQTL analysis, whatever cell type or cell state you're interested in. You can then take these effects, you can compare them, you can try to find shared effects, you can use tools like MASH, and you can take these different cell type specific pseudobulk results and try to figure out where our cell type specific eQTLs are. And so again, all of these papers that I mentioned previously, they do something like this. They do some type of cluster specific pseudobulk analysis. Even if we're able to do this though, there's still limitations if we want to fully leverage what single cell data offers. And so the main difference that comes up now is that some of these eQTL relevant states that we're interested in testing just might not be discrete. They might not be things that we can cluster. I'll give you a couple of examples from work that I've done. I think a lot about T cells. I think a lot about immune cells. And in the world of T cells, one major kind of type of variation we see is cytotoxic versus non-cytotoxic T cells. And these aren't just two clusters. You could try to cluster them, but really if you look at the single cell data, and there have been multiple studies that have shown this, it's more of a continuum. It's a spectrum of the expression of these cytotoxicity genes from low to high. So clustering would kind of disrupt the actual structure of this variation. If you work with something like stem cells that you're differentiating, that's also not exactly a discrete clusterable process. This is more of a continuum, a trajectory. And so for both of these, if you try to do cluster-specific eQTL analysis, you'd probably fall a little bit short of finding the kinds of effects that actually exist. So to do this, we really want some kind of eQTL model that's at single cell resolution. We could dream up some kind of single cell resolution eQTL model. There are a couple of kind of key things it should be able to do, key pieces it should have. So for starters, if we're going to use a regression approach, we need to make sure our regression model is appropriate for the distribution of single cell data. I think this is a really highly contested topic of what this distribution actually should be. There are some people who say, we can normalize and use a linear model. Some people say, no, we shouldn't normalize, we should use Poisson, shouldn't normalize, and we should use negative binomial. There are ways of doing eQTL analysis that people have published that follow multiple of these approaches. I think for what I'm going to talk about today and what we've kind of chosen as our approach is this middle one. So using a Poisson regression. There's a few reasons for this. The first two papers cited here sort of compare Poisson to negative binomial and show that Poisson is a better fit for the single cell data. In our work that we published a few years ago, we actually benchmarked Poisson against linear regressions and found that Poisson was a little bit less susceptible to false positives because of differential expression between cell states. And I'm not going to get too deep into that, but I'm happy to talk about it if people want to hear more about that. But based on some of these different comparisons, we've kind of chosen Poisson as the type of model that we want to use for single cell eQTL analysis. So once you choose your regression model, there are some things we want to be able to model in this. We want a way to account for donor and cell level covariates. Both of these can influence eQTLs. So on the donor level, age, sex, these shape the gene expression profile of a sample. But at the single cell level, things like cell quality, your percent of mitochondrial UMIs, your total number of UMIs that are measured. And so we want a model that can account for that. And then finally, because it's single cell data, we want a way to account for the actual structure in the data. So we have multiple cells coming from each sample. So we have kind of repeated measures We also might have batches. We might have groups of samples that were run together. We might have groups of samples that came from the same cohort. And so we need some way to account for structure. And so the way that we've chosen to approach this is those other covariates we model as fixed effects, and then our batch effects, our individuals, our samples, we model as random effects. So we're able to have both types of variables in this model. So kind of putting all of this together, what we end up with is this Poisson mixed effects model that we proposed in this 2022 paper for single cell eQTL analysis. And to kind of write it out a little bit more formally, this is essentially how we model the eQTLs in the single cell data. So it kind of has our expression, which we're modeling in a Poisson regression. It has this genotype term. So that's really the eQTL. It's telling us how our expression is changing as the genotype, the number of alternate alleles changes. We model all of our different covariates, both at the individual level, but also at the cell level. Pretty standard for eQTL analysis. We include genotyping PCs to account for sort of ancestry, population structure. We include expression PCs to account for biases in the gene expression space. And then we have those two random effects. In our particular data set, we had both donors and batches, but this could be flexible to whatever the structure of your data set is. So from this model, if we want to actually find an eQTL, the way that we assess that is by doing a likelihood ratio test, where we compare the full model to a model without that genotype term. And that gives us a likelihood ratio test p-value that tells us if there's a significant eQTL. So we actually put this to the test in a data set that we had collected prior to this, a memory T cell data set, 500,000 memory T cells from 259 Peruvian individuals, profiled with site-seq. So we had both single cell RNA and protein data. So to kind of simplify our task, we first did pseudobulk eQTL analysis. For each gene, we found its most significantly associated SNP. Then we took that lead eQTL SNP and saw if we could recapitulate it with our Poisson mixed effects single cell model. And so luckily we were able to do this. Here I'm showing the comparison between the genotype z-scores from our Poisson model versus the pseudobulk model. And we can see that largely they're pretty well correlated. We're finding similar effects in both our single cell model and the pseudobulk. So at least at this level, the single cell model is finding the kinds of things we would find in pseudobulk. But keeping in mind that this so far is still a cell state agnostic model. It doesn't have anything that has to do with cell state here. We're still looking only at the eQTL effect kind of across cells more in our entire sample. So how would we need to modify this model to actually incorporate cell state? Well, we'd want to include probably two terms. We want to include a term that just captures the effect of cell state, so essentially the differential expression of a gene based on what state a cell is in. And we'd also want to have an interaction term. That interaction term would be between cell state and genotype, and that would tell us essentially how much that beta for the genotype effect is changing based on a cell's state. And if we want to find significant cell state specific eQTLs, we would look for that interaction beta to be significant, because that would tell us that the genotype effect varies based on cell state. But we still have kind of a big question here, which is how do we represent cell state in this model? We talked about clusters before. Clusters are a pretty straightforward way that in single cell data, we represent cell states. But for the purposes of this eQTL model, it's not really an ideal way to represent cell state. There's a few limitations, one of which I've already raised, which is that we can't represent continuous states with a cluster. There are ways you could take a continuous state, you could bin it into smaller groups and maybe represent the continuous state with these discrete categories in that way. But again, that's not really continuous. And we know that some eQTLs might be specific to a really, really rare subset of cells. So if that's the case, how small would your bins have to be to find that? So we can't really do that with clusters. Clustering always poses the problem of picking a clustering resolution, and previous studies and also roadmaps for how to do single cell eQTL analysis have shown that clustering is such an imperfect process that if we rely on that to find our cell state dependent eQTLs, we're going to miss a lot of things. And a little more philosophically, clusters are sort of one way of analyzing a cell state. It gives every cell one state. We can also think about cell state in this kind of more layered way. And I think there's been both computational work and biological work that kind of speaks to the fact that this might be a useful way to think about cell state. So we can actually think of a cell as representing multiple states at the same time. The first paper referenced here does this with CNMF, kind of decomposing the gene expression profile for a cell into different components that are layered on top of each other. There's also a lot of biological data that shows that cell states can reasonably be thought of in this way because different aspects of a cell state are driven by different transcription factors or different signaling molecules. And so we kind of have multiple lines of evidence here that maybe cell states shouldn't be just every cell has one state, but maybe every cell has multiple states at different levels. So to do this, we need some kind of continuous, kind of additive way of representing cell state. And the way that we propose to do this is using a low dimensional projection of the data. And it's pretty, flexible. You could do this with PCA, do this with CCA, you could do this with NMF. But essentially, you just want some method where you'll be able to get a matrix where for every cell, you have its representation across a series of axes. And then in the sense, if we did PCA, we got PCs, we could consider each PC to be essentially one cell state. And a cell score along each PC represents how much that cell state is represented for that cell. Things that we like about this representation is that it's continuous. PCs are continuous variables. If we use PCA, they'll be independent. Depending on which way you reduce your dimensionality, they may or may not be independent dimensions. But if you want them to be independent, you can choose a reduction method that gives you orthogonal dimensions. And then also each cell then ends up having a representation across multiple PCs. And so in that way, you're able to get this kind of additive approach. Yes.
Yeah. Are you going to talk about how you compute PCs and are there particular difficulties for single cell data and cluster representation, different clusters having different numbers of cells to compute these in a useful way? I assume there are particular tricks and concerns.
Yeah.
Are you going to talk about that at all, or?
I'll allude to that a little bit on the next slide.
Okay.
Yeah, and so the question Ray brought up is, how do we do this in a way that's useful if we really want to find cell states? And so for our data, for the memory T cell data set, we actually had two modalities of data. We had RNA and we had protein. And so instead of using PCA, we used a different dimensionality reduction method called CCA, or canonical correlation analysis. And with canonical correlation analysis, you essentially take observations where you have two different sets of variables measured and then embed them in a low dimensional space that represents the shared variation between those two sets of features. And for us, because we designed our protein space to really cover the depth of cell states for T cells, we knew that these axes would really be biologically interpretable, biologically relevant dimensions for our cells. And so here I'm just showing the first 7 CVs, or canonical variates. And then we annotated each of these CVs based on the genes and proteins that were correlated with that CV. And so with that, we were able to actually annotate these with actual biological functions. And so our first CV, we could clearly see correlated with cytotoxicity genes. Our second CV clearly correlated with regulatory and kind of activation-related genes and proteins. And so we could do this for our first 7 CVs. We used some bootstrapping to figure out how many CVs were the most significant ones. And in the end, these were the ones that we decided were our most relevant representations of cell state for our data set. So kind of going back to Ray's question of here we had this really biologically informed embedding, so we knew that we could get these cell states that were biologically relevant. We've also done this in other data sets where we don't have protein data. Maybe we just have RNA data, we can just do PCA. And when we just do PCA, we're not always able to annotate our dimensions quite as precisely as this. But I think, from my perspective, it's not a huge problem if we're not able to assign a biological function to every axis. We know that what we're doing is we're taking the kind of more complex cell state landscape and just reducing it and sort of deconstructing it into these individual variables. And so while having annotations for each cell state is really nice for interpretation, for the kind of the overall process of finding cell state dependent eQTLs, it's not critical that we actually are able to annotate each of our cell states. And so things like PCA just kind of out of the box work pretty well for this anyway. So we have our cell states continuous. Each cell has multiple scores along each cell state, and we can add these into our Poisson mixed effects model. And so we take our model, we add 2 terms. We add the first term, which is just for our individual cell state effects independent of genotype. And we add an interaction term for the cell state by genotype interaction. And that's really, as I mentioned, going to be our cell state dependent eQTL effect.
Question from Zoom.
Yeah.
Are the low dimensional cell state representations correlated with the sample level gene expression PCs included as covariates?
Yeah, that's a great question. And so there's probably some CVs in this particular example that are correlated with expression PCs, but our CCA dimensions don't really capture anything that's only found in the gene expression space. In that sense, it kind of makes them a little more independent. And then we also do batch correction on our cell state variables. The expression PCs here are not batch corrected because they're just meant to capture, overall bias in the gene expression landscape. But the CVs, because they are, incorporating the protein data and they're batch corrected, it ends up making them a little bit more independent. And so we, again, ran this on our memory T cell data set. We started off with just our, one SNP per gene, the lead SNP from the pseudobulk analysis. We had 6,511 memory T cell eQTLs. When we tested them for state dependence, we found that about a third of them had some kind of cell state dependence in their effect. Again, here we haven't quite figured out what the cell state is yet that they are dependent on. We just know there is some dependence. About 400 of our eQTLs from the pseudobulk analysis actually had a second effect for the same gene. So if you condition on the first SNP, you can find another significant independent effect. When we tested those for cell state dependence, we found that 2/3 of them were cell state dependent. And so we can tell that there really is a substantial amount of cell state dependence kind of hidden within these eQTLs that we just found from pseudobulk analysis. I'm going to show you an example of what this might look like. This is an eQTL for a gene called MAF. This is showing the eQTL across different tertiles of cytotoxicity. So along CV1, our first cell state, you can see that in the lowest end of cytotoxicity, there's essentially no eQTL effect. Here, I'm just plotting all the cells from that sort of quantile lumped together and average to get the individual points here. But then as we increase in cytotoxicity, we can see that the eQTL effect gets stronger and stronger. So this would be a cytotoxicity dependent eQTL. And I think from mapping the eQTLs in this way, there were a couple of just interesting things that stuck out to us. I'm just going to highlight a few of them. There's sort of many more kind of disease relevant and biologically relevant results in the paper, but just methodologically, one thing we noticed is that modeling these continuous single cell states helped us identify more state-dependent eQTLs than if we were to use more conventional categorical or binary classifications. And so, a good example is with cytotoxicity, where if you've worked with T cell data, you're probably familiar with the CD4, CD8 dichotomy of T cells. We conventionally think of CD8s as being more cytotoxic, CD4s are less cytotoxic. Of course, if you look at the actual data, we can see that there's some small subset of CD4s that are cytotoxic and CD8s that are not cytotoxic. But when we did the eQTL analysis, either with CD4 versus CD8 as our cell state or with CV1 as our cell state, we found a lot more eQTLs by using CV1 as our cell state. So we thought a little bit why this might be, what we might be finding here. And what we realized is that we were actually maybe finding more precise states that were mediating eQTLs. And so again, going back to that MAF example, here I'm showing the MAF eQTL with the cells partitioned by whether they're CD4 or CD8. As a reminder, CD4s are less cytotoxic, CD8s are more cytotoxic. The CD8s here are kind of in yellow, and so you can see that the CD8s seem to have a little bit more of a significant eQTL effect. And so this would be easy to just say, okay, great, this is CD4 versus CD8 eQTL. But because we have this other way of representing cell state, we could actually break down each of these into, is it more cytotoxic or is it less cytotoxic? What we find is that within our CD8s, actually, if we look at our less cytotoxic cells, which are the green ones here, there's still no eQTL. So what might have looked like a CD4 versus CD8 eQTL was actually just a cytotoxicity eQTL. And that's not something we would have been able to find just with clusters or pseudobulking. And similarly with CD4, if we look at our more cytotoxic CD4s in purple, they have a really strong eQTL effect. And so what we were seeing in the clusters was just the aggregate of these other cell states that we're better able to model by using a single cell resolution model. And so here, I'm just showing our results from this sort of CV-based cell state analysis. In the paper, we have a lot of other examples because the model is pretty flexible to how you would like to represent cell state. You can use PCs, you can use the CCA dimensions. We also did an example where we used trajectories and computed pseudotime and looked for eQTLs that vary along pseudotime. We can also find eQTLs there, anything that can be represented as a value for each cell. On the genetic side, there were also some other kind of interesting patterns we were able to find here. One is that we found that for some genes, we can actually have multiple eQTLs that can modulate their expression, but that both of those eQTLs could be state dependent in different ways. So this is an example of a locus MDGA1. We know we have two eQTL variants. We know we have a lead effect that's somewhere around the promoter. We have a secondary effect that's further into the gene. Both of them are eQTLs, but if we look at their state dependence, we can see that the lead effect actually becomes stronger along CV1, so as the cell becomes more cytotoxic. But then the secondary effect actually becomes weaker along CV1. So the thing that this tells us is that depending on what state the cell is in, its most relevant or its most active eQTL could be a different eQTL. We can really break this down using these different cell states and this model. The same way you could have multiple eQTLs that are state dependent for the same cell or for the same gene. We can also have, for a given eQTL, multiple cell states that are modulating that eQTL. This kind of goes back to the question before, where we have our baseline eQTL effect, and then each of our axes, so each of our CVs, each of our cell states can move that slope essentially. It can pull the slope up, it can pull the slope down. And so then for a given cell, we know that its eQTL effect, kind of like its effective regulatory impact, is going to vary based on its score along each of these dimensions. And so we were actually able to compute that. So we start off with our baseline eQTL effect. Then for each cell, we essentially multiply its CV scores by the vector of those betas that we computed for each of the cell states. And so that gives us kind of an aggregate version of the state dependence of its eQTL beta. So we can add that to the baseline effect, and that gives us essentially a single cell resolution eQTL beta that we can compute to really see across our single cell landscape, where is the eQTL stronger, where is it weaker. This aggregates all of those different state dependent effects. And this helps us find eQTLs that are present in really small subsets of cells. So in really rare cell types or in cells that have really low expression of a gene that would otherwise just get washed out by higher expressed cell types. So this example is for granzyme A, which is a gene where if we look in pseudobulk, we can see kind of a weak eQTL effect that's present just if we look across all cells. But then if we map the individual single cell resolution eQTL effects, we can see that there's actually some heterogeneity here. There are some cells that definitely have a much stronger eQTL effect, so those more kind of orange, yellow cells. Then there's some cells that actually just have like a negative eQTL effect here, so the completely opposite direction, which are in blue. And so if we sort of partition this out, if we partition out our lowest third of cytotoxicity, so we then went into the betas, figure out that cytotoxicity was the most important interaction here. We look across the cytotoxicity range, we can see that in the upper third of CV1s, our most cytotoxic cells, there's not that much of an eQTL effect. And there's also really high expression of this gene. At the lowest end of our cytotoxicity gradient, there's really low gene expression. But if we zoom in on that, we can see that even with such low gene expression, there's actually a really prominent eQTL there. And so it's a state-specific effect. It's in a really lowly expressed cell type, but it's much stronger than the effect we see in our more highly expressed cell types. So again, we wouldn't have been able to find this if we had just looked in aggregate, because this would not have stuck out. Being able to actually model it at single cell resolution points us to this particular population. And it's kind of biologically interesting because we know that granzyme A is a cytotoxicity gene. And so it kind of is interesting that this variant might only impact its expression in this kind of ectopic way in cells that shouldn't really be expressing cytotoxicity genes. So all of these results that I've shown you are from this one particular way of modeling state-specific eQTLs. It's definitely not the only way at this point to model state-specific eQTLs at single cell resolution. So I want to also just introduce a few other models that have come out over the past few years, all with some small differences from what we've done. I think are just interesting to think about how that might affect our ability to model eQTLs. So the two features I'm going to highlight as I go through these models are specifically the distribution they're using, and then how they're representing cell state. And so we're using a Poisson model. We're representing cell state with these predefined fixed effects. So the fixed effects that we calculate with PCA, CCA, pseudotime on a trajectory, something that we've already computed. Another paper came out a little bit after that called CellRegMap. Their model is a little bit different because they use a linear mixed effects model instead of Poisson. And they represent cell state actually as a random effect rather than a fixed effect. Another interesting feature that they bring in is relatedness, actually modeling the relatedness between individuals in your study, which is really important as we get into these bigger, kind of more biobank scale studies. This year, there's another paper presenting a method called SURGE. With SURGE, again, it's a linear mixed effects model. And rather than predefining your cell states, it actually computes the cell states. It learns the most eQTL relevant cell states directly from the data. So that kind of avoids you having to decompose your data yourself. The model is set up to actually optimize and figure out which cell states best explain the variation in eQTLs across your single cell landscape. So that's another approach to this that's come out. And of course, there's many, many more. Some of them have gotten much more sophisticated, things like Gaussian processes, Dirichlet models. This Gaspacho in particular also learns the latent variables, but is able to learn nonlinear cell states that are associated with eQTLs, which is definitely a limitation of using regressions where we are expecting this linear association. And so they, by using this nonlinear decomposition, are actually able to find spatial patterns too, and look for spatial patterns in eQTL effects. And so it's definitely a space where people are building more and more models, thinking about more and more kind of flexible and creative ways of looking at this relationship between gene expression and genotype. And I think that the kinds of models being developed here are also really applicable to other types of questions. And so there's a lot of other genetic analyses that we might want to do in single cell data where similar approaches could be helpful. One example actually from Shomo's lab that just came out this week is a method called SENT that links open chromatin to the genes that it's regulating. And so here, again, it's a very similar setup, a Poisson generalized linear model. But now instead of looking at the association between genotype and expression, it's looking at the association between a peak's accessibility in ATAC-seq and the gene's expression. And so we can think about the ways that these single cell resolution models can actually get us these really, really precise cell state specific insights into how genes are regulated. There's still a lot of room, I think, to improve these models, though, and I want to highlight two in particular that Jose is going to pick up on in his talk after this. One of them is statistical calibration of the p-values. All of these methods I've presented, or what I've presented, these other methods that have come out, some of them use very different ways of calculating p-values. We use a likelihood ratio test p-value. CellRegMap uses score test to get its p-value. SENT, the ATAC-seq method, uses bootstrapping to get a p-value. We know that the parameterization of these models is really, really challenging. And Jose will get into that a little bit more. And this is something we actually noticed in our own model after a little bit of time, applying it to more data sets. So in our actual paper, we found that we had a pretty reasonable type 1 error when we tested the lead SNP for each gene for associations, for cell state specific associations. Essentially what we did is we permuted cell state across our cells to look to see what the type 1 error was under that permutation situation. But then over time, as we applied this to trickier and trickier loci, we found that there could be actually a lot of cells of p-value inflation depending on what locus we were looking at. So here from another paper that came out of our lab last year, you can see that in one of the HLA genes, if we do permutations, look for cell state specific effects, it's perfectly calibrated. In a different gene, there's a huge amount of p-value inflation. So we found that there actually can be, especially because so many genes deviate from a Poisson distribution or Poisson assumption, we are finding more and more edge cases where our p-values are really not very well calibrated. So that's one thing that we're working on to improve with this model. Another thing is the speed. So all the results I've shown you so far take about 2 minutes per test. This is using LME4 implemented in R with our data set that has 500,000 cells, 259 individuals. But that gets really slow as you're trying to do kind of more complex analyses. We were just testing the lead variant for each gene. If you want to test all the variants in the cis window, this becomes pretty intractable, especially as you think about the bigger and bigger single cell eQTL studies that people are trying to do. Single cell eQTL Gen is a big consortium that's trying to unify, aggregate all of the single cell studies that have been done so far to find these eQTLs across millions of cells. This is going to end up being way too slow for that. And so if we really want to be able to test all the SNPs in a gene cis window, find eQTLs across millions of cells, we need faster models. And so that's what Jose will be talking about in the actual research talk today. But I'll just conclude and wrap up my piece with some of what I think are the more interesting things that we've observed in the process of developing this model. One thing is these Poisson mixed effects models are really able to identify high resolution state-dependent eQTLs, I think at a resolution that we didn't necessarily expect that we would get to. So just kind of like the single cell resolution of measuring an eQTL's effect. And by doing this, we also found really interesting sort of biological findings and really interesting gene regulation dynamics. The two examples I'll highlight is this ability to find eQTLs associated with continuous axes, and also to find these genes where we have multiple independent state-dependent eQTLs that are maybe pulling the gene in different directions. But of course, these models still have a lot of room for improvement. I've mentioned sort of these two ways, the statistical calibration and speed, but of course, there's other things that other people have been tackling, like looking at relatedness, looking at ancestry, looking at tricky loci in the genome. So this is sort of just a starting point, and I think a place from where people are going to develop much, much more sophisticated models.
Today, I'm going to be talking about a new approach to identify single-cell eQTLs that are interacting with cell states following the framework that Aparna presented in the primer earlier this morning. And I'm going to start with a brief introduction, just in case some people just arrived. And the main idea behind these single-cell eQTL mapping studies is that we want to model genetic effects on gene expression from single-cell data. But of course, we can model those eQTLs using the traditional bulk RNA sequencing, where the effect size of a given SNP for a particular eQTL represents the average effect across all the cells in the bulk RNA-seq sample. And this is very useful if this was done in the GTEx project, where we could see that some eQTLs were present in some tissues and not the others. But one of the main limitations about these bulk approaches is that many of the tissues are actually very heterogeneous. We cannot say that they are completely homogeneous, composed of a single cell type. And even within a single cell type, we could have, as Aparna mentioned earlier, multiple cell states that may be actually having very functional and specific behaviors. And this effect size at the level of bulk analysis is going to be biased mainly by the cell type composition. So for example, if we think about PBMCs or blood, what we will see is that most of the eQTLs are going to be biased towards T cells, because T cells are the most abundant cell type in PBMCs. And as Aparna mentioned as well, we have eQTLs that have been found to be cell type specific and also context specific in terms of the cell state. And these first single cell eQTL analysis have shown already that this cell type specificity is present, for example, in the case of PBMCs, where we can see the effect of a given genetic variant on the expression of a gene only present in CD4 T cells, but not found in CD8 T cells. And this is very interesting because it actually tells us that that gene expression for a particular locus is actually controlled in a very mechanistic way that is different between the cell types. But also that difference could also reflect the fact that maybe some loci that are contributing to disease risk are going to act only one cell type and not the other, if those eQTLs are actually colocalizing with autoimmune disease risk loci, for example. And thanks to single cell transcriptomics, and now with other modalities, we can actually resolve the genetic control of gene expression, not on averages of gene expression, but we can actually do it on the causal cells. Therefore, the interpretation of the effect size is going to be with a better resolution and also more biologically interpretable. And just as a reminder, based on Aparna's presentation, this data set that she analyzed as part of her PhD is composed of 259 individuals that are from Peruvian ancestry. 500,000 cells were analyzed, and both the transcriptome and the protein information from cell surface markers that are discriminated for some T cell states were identified. And then 7 cell states were the main focus of this research, mainly in terms of cytotoxic T cells, for example, or even proliferating T cells where we have cells that enter cell cycle. And as you can see in these view maps here, CV1, for example, shows that the cells in the right part of the plot are prone to be more cytotoxic. In the case of CV2, the cells that are in the bottom in purple are prone to be more activated, or maybe they have a T regulatory cell state. And just to recapitulate on the Aparna model that she presented, so she used a Poisson mixed effect model to model the interactions of these eQTLs with different cell states. And by using a Poisson mixed effect model, we use a log-click function to account for the fact that the gene expression is going to be mostly Poisson distributed. We're going to have a main effect that is actually the average or the global genetic effect across all the cells in a population. We have a term for cell state that it's, in this case, it was 7 cell states that are recapitulated in canonical variates. And then we have the cell state interactions between the allele dosage of a given SNP and also the rank of the cells across each of the cell states. But also we have other series of covariates that account for sample level or single cell level sources of variations that are unwanted or not relevant for the identification of the eQTLs, such as age, sex. In terms of the single cells, we have the total number of reads found in that particular cell. We have the mitochondrial expression. We also have expression PCs that are mostly uncorrelated with the cell states. And we also have genetic PCs to account for genetic ancestry. And importantly, we have also these random effects that account for the fact that we have multiple cells belonging to a given individual. Therefore, we have a covariance structure in the data that we have to account for. And also, we can add a random effect for the batch, because in these experiments, not all cells can be processed in a single experiment. We have to run multiple experiments of single cell RNA sequencing or CyTOF. And usually, for example, you would pull around, let's say, 7 or 13 individuals in a single experiment or a single batch. So I'm going to be talking about some of the challenges to perform this context-specific eQTL mapping. And the first one that Aparna already mentioned is that most of the assumptions that we would assume using a parametric approach would be violated, because the distribution of gene expression is very variable. Even if we think about a very homogeneous cell type, when we compare between genes, some of them are going to be maybe more Gaussian, some of them are going to be more Poisson, some of them have a very irregular shape. And therefore, for most of the genes, actually, the Poisson model works very well. But there's going to be a set of genes where the interactions are going to be actually false positives. And we actually don't want that, of course. And the other issue is that, as Aparna mentioned already, these methods are not very scalable, because fitting a linear mixed model using that many cells, and now we are generating data sets that are millions and millions of cells and almost thousands of individuals. That's actually very impractical, particularly when we think about doing genome-wide cis testing. So not only focusing on interaction of the lead effects, but also doing a whole scan across a particular loci. And just to emphasize this part, there's actually no consensus to have a model that resolves the parameterization of single cell gene expression. Initially, when the droplet technologies for single cell RNA sequencing emerged, a zero-inflated negative binomial model was proposed, mainly inheriting this idea that bulk RNA-seq is negative binomial distributed. And they added a zero-inflated term to account for the fact that we have many zeros on single-cell data. Then there were other papers that actually kind of made a point that a Poisson regression was actually better. And that was proposed 2 times, actually, so far in, let's say, the history of these sort of papers. And for example, from Satya's lab, they proposed a method called SC-transform, which essentially models the distribution of gene expression for all the genes using a negative binomial regression, but then regularizing the parameters depending on the level of expression of the gene. So the takeaway message of this is, of course, it's very complicated, and also it's a sort of emerging and very dynamic area in the single cell area. And first, I want to introduce a biological example that I think It's very interesting, and I'm going to be focusing on this gene for the rest of the talk. So this is a gene called CTLA4 that is a negative regulator of T cell activation. It's actually a cell surface protein that is expressed on T cells, and this protein is actually called CD152. And the expression of this particular gene is larger in activated T cells or Tregs. And this gene has been associated to multiple autoimmune diseases such as rheumatoid arthritis or multiple sclerosis, and also even with a variety of cancer types. And the reason why this gene is very important in terms of the biology of T cell activation is because this gene is actually located in the cell surface protein, in the cell surface of the cell, sorry. And once once an activation signal is triggered by an antigen presenting cell, like the cell showed here on green, which could be a dendritic cell, the activation happens via the TCR receptor from the T cell and the major histocompatibility complex. And also stimulation happens between CD80 and CD28. In the case of the activated T cells, there is going to be expression of CTLA4 that is actually going to bind to CD80, and then CD28 is going to be, let's say, less bounded to CD80. That actually makes a functional inhibition of the T cell signal, and this is a natural mechanism by which T cell activation is regulated. And for this particular gene, if we think about mapping at the cis region, let's say 1 megabase upstream and downstream from the transcription of star site, and if we focus on the data set that we've been talking about, a boson mix effect model would take around 2.2 minutes in average. And if we were to map the entire region, this would represent about 4 days, which doesn't sound that crazy if we can parallelize this. But if we want to do this for all the genes in the genome, we want to have a more scalable approach. And therefore, as I mentioned, focusing on elite variants is possible, and you can have very important insights. But maybe there are some interesting signals that you're missing that are maybe not the lead variants, but maybe secondary or other variants that are not captured by the main effect. And as you can see, if we wanted to apply a non-parametric approach, such as bootstrapping or even permutations, this is practically impossible because of the computation time limitation. So what I'm going to talk about is a new approach that we claim to be robust and scalable in most of the instances to map context-specific eQTLs. And what we want to do is tackle these two issues that I presented. And to solve, for example, the statistical robustness or this lack of parametrization of the data, we're going to use, instead of non-parametric approach via bootstrapping, to derive empirical p-values instead of relying on the analytical assumptions. And of course, this is going to be even more slow if we think about the actual computations. And because we want robust p-values, we need more scalability. And the way we do this, and I'm going to explain each step more further, we do a residualization of the shared fixed effect covariates for any given test. We implemented the linear mixed modeling now in Julia instead of R. And then we do some sort of cell aggregation via Poisson desampling, this to reduce the dimensionality of the data and speed up the computation. And the idea is that now the p-values are going to be calculated for an empirical bootstrap distribution. And the approach that we use is a very standard bootstrap approach, which is for each SNP for a given gene, we're going to sample the cells with replacement, generating a new bootstrap sample. Then we're going to compute the betas using a linear mixed effect model for the main interaction, for the main term and interaction terms. And then we're going to derive a p-value for each of the terms under the null hypothesis that the beta value is 0, because we are testing whether there's an actual effect, whether it is in an interaction or main one. And this is an example of what we see. And this is actually a very extreme example where we have the expression of the gene CD8b that is associated to more cytotoxic T cells. And what you see in the x-axis is a p-value derived from a likelihood ratio test without doing any bootstrapping procedure. And what you see in the y-axis is a p-value derived after bootstrapping. And as you can see, there are extreme scenarios where the p-values are actually very inflated for the likelihood ratio test. And even in the zone of interest, that I call it like that, to reduce the level of false discoveries, we focus on this region. We see that we're going to have some p-values that are very different between the likelihood ratio test and the p-value from the bootstrapping. And therefore, by doing this, we are going to reduce the false positives from our identification of eQTLs. But even doing bootstrapping alone is going to be computationally expensive, because we are pretty much going to do n number of bootstrap replications for every single SNP for all the genes in the genome. And the precision of the p-value is going to depend on the number of bootstrap samples that we compute. So let's say if we want to have a p-value that is, let's say, 0.001, then we will need to perform 100 bootstrap samples to get that p-value. But we can make this observation that not all the SNPs are going to actually be significantly interacting with the cell state. Some p-values are going to be not that small. So therefore, for those SNPs, the p-values could be derived from a lower number of bootstrap samples. And the way to determine that is using an adaptive bootstrapping procedure, which is very simple. And in this schematic, you can see 2 SNPs. And in, let's say, the x-axis, you can see the number of bootstrap samples that we would perform at every single step. And what you can see is that if we were to perform 100 bootstrap samples and then compute the p-value, we would see that in some instances, we actually don't need to go up to 100,000 bootstrap replicates, because at every single step, we test the null hypothesis, and we actually empirically ask whether the beta values for any of our parameters of interest are larger than 0 or lower than 0. And if there's at least one observation that actually crosses 0, either from the left side or the right side, then there's evidence that we have a given p-value at this given stage, and we therefore stop the bootstrapping iteration. So in the case of the SNP2, for example, we needed more bootstrap replicates to actually derive a p-value that is smaller. So now the n number of bootstraps is going to be pretty much defined by the limitation of our computing resources or even of our purposes. And this is an example of the gene CTLA4, where, for example, if we perform 5 rounds of bootstrapping to derive p-values that go from minus log 10 from 2 to 4, we would see that at the first stage, we have some genes that actually we don't need to bootstrap anymore, but then we can go deeper. And by doing so, we actually, the takeaway message from this is that from the 2,500 SNPs that we were going to test, at the 5th round, we only have left 1,600, which means that we actually don't have to keep doing bootstrapping. Therefore, we reduce the computation time. So this adaptive bootstrapping approach has been used before, for example, in the SEN work that Aparna mentioned from our lab, or even in matrix eQTL, which is used for bulk RNA sequencing. And one other step that we can take is actually residualizing the gene expression in terms of the covariates that are actually constant in all our testing procedures. And remember that we have a series of covariates that are going to be constant for any given SNP and gene pair. Age, sex, the number of UMIs, mitochondrial expression, and the genetic and expression PCs is something that is constant across all the testing for all the SNPs. And the rationale behind this is that we can actually regress out that using a Poisson model. And then we will have corrected gene expression in the form of residuals. And because of this, now we can actually focus on the terms that we are actually interested in. So we can model now the residuals as a function of the main effect, the cell state, the interaction, and now including, of course, the random effects to account for the repetitive measurements. So this residualization is actually relevant because conceptually, we actually don't need to focus on those terms. Those are things that we actually want to account for. But also in terms of the computation time, if we were to map the entire region for CTLA4, what we would see is that there's a considerable improvement in terms of the speed. These results are shown for only a likelihood ratio test, but you can imagine if we were to do bootstrapping, this actually could help us a lot to actually have better results in terms of the speed. And one other thing that we tried was, instead of using the LME4 library that is implemented in R, we switched to Julia, which has a very nice implementation of mixed effect models. And this library, which is called mixed models, considerably outperforms the R library in terms of the speed. So again, if we were to focus on this particular gene, CTLA4, we would see that to map the entire region, it would take around 15 hours. But now, if we were to do it in Julia, in getting exactly the same results, even at the level of decimal points, we would actually do it in 1.8 hours. So that's actually a huge improvement, just switching from a programming language. And one of the cool things about this is that the maintainers and the creators of these two packages are the same people, which implies that the actual algorithmic implementations are very similar and also forced to have pretty much the same results to replicate pretty much between programming languages. And one step further that we can take is reducing the dimensionality of the data. So for this, we would like to, of course, reduce the number of observations to reduce the computation time. But also, we would like to maintain the structure of the cell states and all the covariates in the data. Because in the end, what we would like to do is feed in faster these models, but also maintain the power to detect the eQTLs that we could potentially find. And one of the things that Aparna and a rotation student called Mika did in the last year was trying to see if we were to aggregate cells into smaller, let's say, pseudo cells, whether we could actually recapitulate the results that were found in the main paper using the Poisson mixed effect model. So in this case, in this plot, I'm showing the number of eQTLs that had an interaction for any given cell state. And one simple approach or baseline that we can take is we can just downsample the data and hopefully maybe we could recover the same number of eQTLs. And the answer is yes, but you actually lose power. So in this case, instead of capturing almost 100% of eQTLs, we are capturing pretty much 75%, which is not bad, but we are losing power. So one other approach is, instead of doing a random downsampling, we can actually start aggregating the cells. And the approach of this is creating pseudo cells, where the idea is that you pull the reads from every single cell that are similar across a given cell state. And in the simple terms, if you were to take a single cell and take the 10 nearest neighbors, you can aggregate those 10 cells into one. And then by doing so, you reduce the dimensionality of your data into a 10-fold. And what Mika and Aparna saw was that once you do this pseudo cell aggregation, you can actually recover pretty much all the eQTLs that you would find using the single cell model. So this is a huge reduction in terms of the data size, because we start from 500,000 cells, and now we are actually having a data set that it's only 50,000 observations. So you can see why this aggregation method is very powerful. And the general idea of this Poisson sampling is that if we were to take a random sample of let's say, two-dimensional parameter space, we would see that with a random sampling, there's going to be regions in our parameter space that are going to be empty just because of the stochastic nature of the sampling. But with a Poisson sampling, the general idea is that we want to have a more uniform representation of the parameter space. And in the case of the eQTL mapping, what this is going to do is that it's going to capture the cell state, even in instances where we have, for example, a very small population of cells in a specific position across the cell state. And this is the theoretical explanation why this method is actually performing better than doing a random sampling approach. And now, if we go back to the identification of dynamic eQTLs for the CTLA4 locus, what we will see is that using single cells, as I mentioned before, it would take 1.8 hours. But now using the Poisson disk, it's only 2.4 minutes, which is a huge improvement. And you may wonder, well, how accurate the results are. Like, so we, at this stage, have used a series of procedures such as residualization, and then the cell aggregation. And if we were to compare, for example, the set scores between the residuals model that I'm calling here and the posomics effect model, which are the results that Aparna got in the original paper, you can see that for the main effect, which is the plot on the left, we actually have a very good correlation and concordance of the set scores across many genes, which is the genes that are actually interacted with CV1. But then if we look at the actual interaction, which is represented on the right, we see that there's still a large concordance and also correlation between the estimates. And one interesting thing is that most of the discordance that we observe, at least for this particular case, is that we have some instances of genes that have nonlinear relationships between the main effect and the interaction, which implies that maybe using the residuals approach and the posomics effect model, we're going to have some discordance, but we are still actually capturing the same eQTLs, which is very encouraging. Maybe the set score is a little bit off because we can model a nonlinear trend using different linear approaches, but in the end, we are actually recovering the signal. And one application of this method, for example, in the case of CTLA4 that I found very interesting, is that if we were to look at the main effect, we can see it here in the upper plot. We see a huge spike in the signal of association in the middle of the plot, which actually corresponds to the transcriptional start site in the promoter. And the gene is actually located downstream this region. And from Aparna's analysis, there were two main effects that were independent that are actually located in the promoter. And those, well, one of those lead eQTLs actually is interacting with CV2, which is a cell state that is explaining T cell activation and Treg status. So this is exciting because this gene is actually highly expressed in Tregs, and the fact that we have a signal there is actually encouraging because this signal is actually colocalized with an autoimmune disease risk loci. But then if we were actually to look another cell state, not T cell activation, but if we focus on cytotoxicity, we see another signal that is actually independent from the primary and the secondary lead variants that were found before. And this independent signal is actually not a main effect either. And I think this is very interesting because this could reflect the power of using these context-dependent eQTLs, because maybe most of the eQTLs that you would find that have some sort of interaction, you can find them from single bulk analysis. But what this is potentially showing is that some of those signals are going to be missed because of the heterogeneity of the cell types. And therefore, you can actually recover those effects using a single cell model. The other thing is that one possible explanation is that we have an effect in this particular region because maybe the main effect is not present because we have, let's say, a cross effect across interaction, which means that probably the overall effect is canceled at the level of the main effect, but the interaction is capturing better that relationship. So I'm gonna conclude with some remarks about this talk. So the first thing is that we know that these context-dependent eQTLs are hard to model, and mainly because they suffer from a statistical robustness and they are not scalable. What we propose here is a new method that could potentially be scalable, and by doing so, we can derive empirical p-values from a bootstrap distribution. And the speed is tackled by using a simplified linear regression framework by residualizing things that we are not interested in. Then we implement our approach in a faster programming language implementation. And we also aggregate cells into a lower dimensional space while preserving the power. And finally, I showed an example where we have a context-specific eQTL for CTLA4 that modulates expression of this gene only in cytotoxic T cells. And the identification of this particular eQTL is actually done in an unbiased manner, which is actually very important to mention. And the interesting part here is that there is no main effect detected at the pseudobulk or single cell level for this particular eQTL.